Good morning, students. I, Miss Madhvi Singh, your science teacher. Today we will do human digestive system. See here, this is human digestive system. We will learn how it works. Let's start. This is your rectum. Next, large intestine, small intestine, pancreas is there, stomach is there. In case of digestive system, next we are having liver as the gland, esophagus is there. The human digestive system starts from the mouth and ends to the anus. There are different type of glands, salivary gland that is present. Now see how it works. When we take any food, with the help of the teeth, the mastication and crushing of the food take place, and we are having salivary glands, and the food that we take are having carbohydrate, and in the salivary gland we are having salivary amylase that acts on the food particles, that is carbohydrate, and digest the carbohydrate into simpler form. Now the food is being converted into bolus. This bolus now moved through the esophagus or the food pipe, and in the esophagus, the lumen of the esophagus, as you can see over here, there are large number of layers which makes the esophagus, that is tunica adventitia, stratum circulare, stratum longitudinale, tila submucosa, lamina muscularis. These are some of the layers through which your esophagus are made. So in this esophagus, the inner two layers that do peristalsis movement. See here, this is peristalsis movement. This is how the food enters into the stomach. The rhythmic contraction and the relaxation of the esophagus to pass the food into the stomach, that movement is called peristalsis movement. Now the food, that is the bolus, has reached in the stomach. The stomach, as you can see, are further far more divided. Fundus region is there, cardiac and body parts are there. The stomach is a sac-like structure, muscular sac-like structure, C-shaped structure. Inside this, you can see the stomach is having large number of foldings, that is called rugae. So in this, they are having large number of gastric glands which secrete gastric juice, and this gastric juice are very much acidic in nature. To protect the stomach from the gastric juice, we are having mucus secretion or mucin coating over the stomach. This you can see there, these are the gastric juice that make the stomach highly acidic medium. The stomach and the esophagus both are separated. The esophagus throw the bolus into the stomach. These are bolus which are released into the stomach. Now in the stomach, this bolus remains there for 4 to 5 hours. Now in the stomach, there are large number of gastric juices, that is hydrochloric acid, pepsic, intrinsic factor, lipases, which allow the digestion of the food, and with the breakdown and allow the food to break down into smaller forms. The role of the HCl in the stomach. The HCl used to digest the microorganisms that is present in our food. It helps to kill the microorganisms. Next, this HCl also convert the inactive pepsinogen into pepsin. With the action of the HCl on the pepsinogen is being converted into the pepsin, and this pepsin helps to break down the protein into simpler forms, that is amino acid. See here how beautiful amino acids are being released, and these are further absorbed in our body. Now there are some intrinsic factors that help in digestion of vitamins B12, because these vitamins are digested with the help of these intrinsic factors. This intrinsic factors comes and binds on these vitamins and helps to break down into simpler form that can be absorbed by our body. This is how the digestion of vitamins takes place inside our body. Now the digestion of the fats. The digestion of the fats takes place with the help of lipase enzymes that is present in our stomach. This lipase enzymes helps to break down the fats into simpler forms. These fats are converted into simpler forms, and the simpler forms, that is free fatty acids, are being absorbed by our body, and further digestion of these fats takes place inside our small intestine. You can see the beautiful diagram of the stomach. Inside this stomach, when the food stores for 4 to 5 hours, the stomach contracts and relax. Now the food, that is the bolus in the stomach, is not called bolus, it is called chyme. It allows the medium of the food to change into acidic environment, that is called chyme. Now this chyme are released through the pyrolytic sphincters that is present at the end of the stomach. This pyrolytic sphincters allow the chyme to release into the duodenum. Just you can see here, the pancreatic juices are also released over there. This pancreatic juice contains large number of pancreatic enzymes that are responsible for the digestion of the food. For example, in this pancreatic juice and the bile juice which are released from the liver, they contain hydrogen carbonates. This allow to change the acidic environment into the alkaline environment and makes our food now alkalinic medium. Now see, trypsinogens. These are the enzymes that are responsible for the breakdown of proteins. This trypsinogens allow the bigger form of the protein to get converted into the smaller form, that is amino acids, and these amino acids are being absorbed by our body. So in this way, the digestion of fats, proteins takes place inside our body. You can see the beautiful presentation over here, how the fats, the lipids, the proteins, the carbohydrates are being digested inside our body. This is the digestion of fats. This is how the fats are being digested with the help of enzyme called lipase. This is how the digestion of fats takes place, and they are being converted into the free form, monomer form, which are being absorbed by our body. Now next we are having, as we move further, we are having the accessory glands, that is liver and the pancreas. The liver and the pancreas, the liver is the largest gland in our body. This liver secretes an organelle that is called gallbladder. This gallbladder has secretion of bile juice. This bile juice helps to emulsify the fats, that is called emulsification of the fats. Now as we move further, we are having duodenum, that is small intestine and large intestine. The small intestine is further divided into duodenum, jejunum and ileum. The duodenum is the U-shaped area where the digestion used to take place. Just inside that, if you will observe the internal view of the small intestine, you will observe large number of highly fold convoluted area, and in that convoluted area there are thousands and thousands of microvilli that is present, that are hair-like projections that increase the surface area. And within this, if you observe, this hair-like projections are rich in blood vessels. This blood vessels, this microvilli helps to absorb the minerals, the monomers from the food and directly send this into the blood vessels. That's why they help in maximum absorption within our body, and the nutrient that it absorbs from the small intestine are directly passed into the blood, and the body takes the nutrition in the form of nutrient they are taking. Transfer of ions are there, transfer of vitamins, minerals, carbohydrates. The carbohydrates are being converted into the simpler form, that is glucose, by the process of glycolysis, and further breakdown takes place. The chylomicron, that is digestion of the fats. The chylomicrons are triglycerides. They are absorbed in our body. This is called digestion of fats. They are in the form of chylomicrons. This small intestine helps in digestion of fats, that is emulsification of the fats. The bigger fats are being converted into the small intestine. You can see over here there are large number of highly convoluted hair-like projections inside the small intestine. This is called microvilli. They are rich in blood vessels. They help to increase the surface area of the small intestine and make for maximum absorption of the food. This is how your small intestine looks if you observe it under the microscope. As you can see over here, that this is the longitudinal section of your small intestine, and there are large number of microvilli present. The calcium that are being absorbed by our body and other nutrients that are being absorbed by our body, such as zinc, vitamin B12. Vitamin B12s are digested with the help of intrinsic factors. Bile acid, that is bile juice, release some chemicals, some enzymes, that is called bile juice, that are rich in bilirubin and biliverdin. So in esophagus, what is happening? Peristalsis takes place. See here how beautiful representation is this. This is called peristalsis, rhythmic contraction and relaxation. Your esophagus contract and relax in a segmented manner, and the food are being passed into the next. In this way, the digestion used to take place. And you can see over here, when the small intestine ends, there are large intestines. At the junction between the small intestine and large intestine, we are having a pouch-like structure that is called vermiform appendix. Now you see the structure of the large intestine. This is just the internal view of the large intestine, how your large intestine looks if you observe it carefully under the microscope. And in the vermiform appendix, there are large number of bacteria, as you can see over here, that is E. coli is present in vermiform appendix. Vermiform appendix is a vestigial organ. This bacteria helps in digestion of roughage inside our body, so it's very useful. Now move on to large intestine. The large intestine is ascending colon, transverse colon, that is three segmentation is there: colon, rectum and anus. Ascending, transverse, descending colon. The lower portion, that is called descending colon. And next, if you move to the downside, next we are having a pouch-like structure, that is rectum, that is used for temporary storage of the food, and opening is called as the anus, through which excretion passed out. Thank you.